Thank you everyone so much for attending this February's webinar from the GDC. My name is Bill Wysocki. I am the GDC User Services Lead, and I'm here with my colleague Tara Lichtenberg, who is the GDC Clinical Data Curator. Today, we're both going to talk to you about the TCGA resources that are available in the GDC. We know that's a fairly popular topic, so why don't we get started? First, I'll go through our agenda very briefly. We're going to have Tara introduce the TCGA program, as well as how the TCGA clinical data is collected and made available in the GDC. After that, I'm going to give a small introduction to what molecular data is available from TCGA in the GDC, as well as give a few small demos on how to retrieve TCGA data from the Genomic Data Commons. With that, I will hand things over to Tara.
Thanks, Bill. I'm going to give a very brief, if you go to the next slide, overview. I think most folks who are calling in probably have an understanding of what TCG is and was. But this graphic created by the project team at NCI does a good job of explaining the players involved in TCGA. And I think most people understand that it was a huge collaboration among hundreds, if not probably over 1,000 individuals who made the results of TCGA happen to sequence and characterize data from samples that were collected from, they came from over 11,000 cases, which represent patients. And then the majority of them have both a primary tumor and a matched normal. And there's 33 specific cancer types that were focused on for TCGA. And this project began in 2006 as a pilot, continued on to a full-scale project in 2009, and data and samples were, we stopped collecting them somewhere in 2015, but the work still continues as proof that there's interest in this call. So if you go to the next slide, my goal here today is to explain the clinical data, but I wanted to at least point out sort of the process of how the data was moved and collected. And so the three primary entities within TCGA that created data that is now available at the GDC are the tissue source sites, which provided the samples from the patients. And they also provided clinical data to go along with those samples and information about metadata about the samples themselves. The BCR collected both the samples and the clinical data. They did QC. So they both cut slides from those samples, reviewed them to ensure there was enough necrosis or not too much necrosis and enough tumor nuclei, and then ensured that they extracted the analytes, and they ensured that the analytes had enough yield and were of good enough quality for sequencing. And then all of this was passed on to the sequencing centers, which is the primary data that is housed at the GDC now. Of course, Bill will go through the GDC's pipelines and how that data is presented on the GDC specifically. But I just want to make sure that folks understand that there were a lot of people involved in this process, and it wasn't it was sort of a messy business, and so there was a lot of work to clean up not only the process but the data itself. So if you go to the next slide, I'll jump into the clinical data. So I'm going to do like a why, what, when, where, how of the clinical data. The data process started with what we call disease working groups. And there were 33 different disease working groups, one for each study. And the studies represent the tumor types that you see on the GDC's site when you're able to filter down. So there were a lot of people, and each group had its own diverse group of experts. So there were folks like surgeons and oncologists and bioinformaticists and geneticists, all kinds of folks who had at least one thing in common, in that they had an interest in the specific disease that was being studied and some level of expertise. And so it was each of these groups that came up with the questions that were collected on the clinical forms. So the positive side of this is that every disease has really nice disease-specific information included. The downside was that every disease has a very specific and unique set of questions. So for example, if you wanted to do a cross-study analysis on something as simple as height and weight, there were some studies like the endometrial studies and the gastric studies that found that information to be very important. And so they included it on their forms, but many of the studies didn't find something like height and weight important, so it was left off the form. So that's sort of the downside. But there is a lot of great information available. We just need to know how to get to it. So if you go to the next slide. So what was collected and when? So the disease working groups came up with every disease working group had to come up with questions for 3 different forms. The first one was a sample submission form, and that was sent with the sample at the very beginning when the samples were sent, and many of these were filled out and never made public because the sample didn't qualify. And so we didn't really consider the case to be enrolled until it passed those QC checks. So when I explained that the BCR cut slides and reviewed them for necrosis and tumor nuclei and did the QC on the analytes, it wasn't until the cases passed that step that we requested an enrollment form from the site. So you can imagine anyone who's done clinical curation out of an electronic health record or any other kind of medical database, it can be really difficult and time consuming to curate data. So we didn't want to force them. In the beginning, some of the pass rates were pretty low, so we didn't want to force them to, curate all of this data for samples that wouldn't even be sequenced. So we waited until they were qualified and they filled out this enrollment form, and that's where you'll find, so the sample submission form includes a lot of nice metadata about the sample itself. The enrollment form is where you'll get a lot of the patient-specific information like stage and grade and additional demographics about the patient. And all of that disease-specific information that the disease working groups came up with, for the most part, is on the enrollment form. And then a year after that enrollment form was collected, we requested a follow-up form. And the follow-up form served for 2 purposes. There's a section on it, and it's also on the enrollment form, that captures information about recurrence and progression. It's called the new tumor event. It also captures information about new primary tumors that the patient may have been diagnosed with. And the follow-up form served another purpose in that the disease working groups often discovered, once the sequencing data started coming out, or just as time went on, they realized that there were elements that they didn't include initially in that enrollment form that they really wanted to collect. So rather than adding it to an enrollment form and only getting it from the patients who hadn't completed one yet, we put it on the follow-up form in hopes that people would be able to submit those data for more cases. And so sort of the changes over time are why you might see variation within the data set. But there also, I bring that up because there's this confusing aspect of follow-up forms, which is the versions. And so you will see the versions when you look at them, and I'll talk about the bio tabs and the XML in a little bit, but when you look at them, you'll see many different versions of the follow-up forms. And that doesn't mean that every case has one of those versions. Those versions represent changes over time to that follow-up form. So whereas one case might have 2 instances where we got 2 years of follow-up out of a case, they might have 2 follow-up forms of the same version, and it might be an earlier version, or they might have 1 earlier version and 1 later version. So when you're looking at the follow-up data, you need to look at it in its entirety and realize that there can be more than 1 follow-up form per case. We ran into some issues with that data collection that we had to build some QC metrics around, where people were showing up as deceased on the enrollment form and then alive on the follow-up form, and it got really crazy. So we've cleaned that up, but if you see any issues where you're confused, hopefully we can walk you through it. But just keep in mind that those versions represent changes to the project itself over time. Then in addition, there were 3 forms that were not unique to the study, and that's the other malignancy form, which actually has its own XML file. This was really important at submission because we wanted to make sure that we didn't include cases with prior malignancies where the patient may have been treated with something that could have modified their genetic profile. And so we did not, for the most part, we didn't allow prior malignancies, but they filled out this form because there were some treatments that we accepted. And so you'll see there for qualified cases what prior malignancies they did have. And many of them didn't receive systemic treatment. So that was a way for us to confirm that they did not receive systemic treatment that would have affected any kind of genetic variation. And so, or caused any genetic variation. And so we allowed those, but that's can be helpful information that folks don't know. A lot of folks don't know is out there. And then the treatment form. So I wanted to point out that the pharmaceutical form and the radiation form, which the pharmaceutical form is called the drug form is called the drug section within the XML. They were 100% optional. And we get a lot of frustrated users who hate that there isn't more treatment information. And I would just sort of flip that on its side and just be really grateful that we got what we did get, because what we did get was 100% voluntary. And so I'm very grateful that folks who did submit that data took the time to do so. So you go to the next slide though. So continuing on with what and when, there are 900 unique TCGA data, over 900 unique TCGA data elements in the data set. And that's across all 33 studies. But we've had recent discussions about should we index more, should we not? And I think that's still to be determined. But just to let folks know, there are some questions out there that were so specific and part of some of the really rare disease, the rare disease studies of the 33 that only a couple cases answered the question. So while it may be really important to a couple people in this world, we don't know if it warrants the level of curation to bring that into the GDC. So these are discussions we're still having. But just so you know, if you want all of that data, it's available in the bio tabs. It's available in the XML, and it can be parsed, any way you want it to be. But when you look at the GDC, we've indexed a core set of data elements that are included in all or nearly all of the 33 studies so that you can get a rich sort of depth of data for each of those elements. And like I said, stay tuned, we might end up indexing more, but that's still to be determined. If you go to the next slide though. So where and how? So the data were collected, like I said, by the BCRs, and there was a lot of communication between the tissue source sites and the BCR to make sure that what was submitted was what was expected to be submitted. There were a lot of validations put in place because, and enumerated fields were added because of the early on, what we received early on. A couple of things I really wanted to point out was some of the tissue source sites had a lot of access to the electronic health records, but not a lot of bandwidth. And some of the sites had a lot of bandwidth, but they might have been like commercial biobanks where they didn't have much access to go back to the treating site or the site who had direct access to the patient to get more data. And then there was a bunch of sites in between there. So if you see missing data, oh, and I'll talk in a minute about what was required and not required. But if you see missing data in there, it's because sites were only obligated to provide a subset of the data, and everything outside of that was voluntary. And again, like the treatment, we should be grateful that some folks spent the time to provide extra data. Other folks whose primary goal is to treat patients didn't have a whole lot of extra time to be curating data. And then I also point out that this was it started 15 years ago. So the original forms that were used for TCGA were carbon copy paper forms. So any really young people on the call may not even remember or know what that looks like, but it's like the pink and yellow behind the white where you fill it out and it writes through. So we were dealing with that in the very beginning. And so those forms had to be entered into our electronic capture system, Open Clinica, which was up and running in 2009 when we started collecting clinical data from on the larger scale when the full scale project started. But just to warn people that if there are nuances within the data, Things in the data collection world were very different 15 years ago, and even prioritizing this type of data, the clinical data there was a different mindset back then. So if you have questions, let us know, but there are some times when we just have to say, yeah, that's not available, and we're sorry. I did want to point out, both on the required side and just access to the forms that were collected in Open Clinica, there are PDF versions of what was included in those electronic forms. They're currently housed at the BCR still, so I put the link there. And on those forms, you'll see an asterisk next to the required questions. So if you have any question about what was required or what wasn't, sort of what data you should get more depth to, look at those tagged questions and you'll know what was required. If you go to the next page. And then where. So the data was captured, as I mentioned, in Open Clinica. And once the data was completed and QC checks were performed on them, our bioinformatics team transformed them into XML files, which is what they were originally submitted to the Data Coordinating Center, which was then turned over to the GDC. And so now all of those XML files are available at the GDC. I mentioned that there's 2 for each patient. That's the biospecimen and the clinical. Biospecimen includes information about the DNA and RNA that was extracted at the BCR, as well as a little bit of information about the slides and some metadata about the samples themselves. And the clinical has all of that information, all of that clinical information from Open Clinica, including the enrollment form, the follow-up form, and the other malignancy form actually has a separate XML file. So you'll have to look for that on the GDC. It'll be on its own. But it does also have a bio tab. So if we go to my last slide, which is about bio tabs, early on in TCGA, it was decided that not everyone can work with XML. And so a lot of the clinicians wanted a flat file that they could see all the cases for a single data set and the corresponding data. And so we created what we termed the bio tabs. And I wanted to make sure everyone knows that everything in a bio tab file, it's a text file that can be viewed, as a text just in a text editor, or you can view it in a spreadsheet like an Excel sheet. Every field in that file comes from an XML file. So the bio tabs that are clinical in nature, you'll see the table below. The form name, if you want to go to that website where the original forms are housed, the form name is on the left and the bio tab name is on the right. And the bio tab name also represents the sections within the XML files. So the XML files were basically split out by section, and these flat files were created for each section. Now there is some, I don't know if it's helpful or not, but there is some data that you lose when you use the bio tabs. There's a procurement status, for example, for every data element within the XML that you'll lose when you look at the bio tabs. But for the most part, you're getting the majority of the data. You're getting the question that was asked and the answer that was provided. And those are available on the GDC, and Bill will show you how to get to those during his demo. But I just wanted to point out that if you do have the ability to parse the XML, you're not losing anything by using the XML files. And so I think that's everything I wanted to say, Bill. But
all right, thanks a lot, Tara. And I am now going to introduce some things about the TCGA molecular data, as well as a few points about what data in TCGA is available in the GDC and how it's available. So here I put together this general flowchart for how molecular data enters the GDC and how it's processed. So on the left here, we have the original TCGA molecular data. This is colored orange because this isn't something that's directly accessible for end users. And the original TCGA molecular data was taken in by the GDC harmonization pipelines and processed into the TCGA data that can be found in the GDC data portal. That data, certain types of data were processed into math files, and those files are where the mutations are found in the exploration portal. So that's mostly whole exome sequencing right now. There's another pipeline that is more or less based on the researchers, and that's data that goes into the GDC publication pages. And I'm going to go into most of these aspects a bit more, but I'm just introducing the flow now. So to introduce TCGA molecular data harmonization, I just want to go into the main purpose of the GDC, which if you've been watching these webinars, you've probably heard many times. The purpose of the GDC is to harmonize molecular data across standardized pipelines. And for this reason, TCGA was processed no differently than in any other program. The goal was to process it just as we would process any other program. One nuance I would like to bring to everyone's attention, though, is that TCGA was one of the projects that was released by the GDC at initial launch. So TCGA was one of the first, when I say projects, I mean programs that was launched when the GDC first came to be. So I'm just going to go quickly over what kind of data is available for TCGA. We have whole exome sequencing, RNA-seq, microRNA-seq. For all of these, we have alignments, all of which are aligned to the same reference genome, which is GRCH38. In our reference file section, you can see the exact version of that. But for whole exome sequencing, we have the variant calls that were called using 5 different pipelines, variant calls that are annotated, and then finally the variant calls that were originally VCFs were converted to MAF files. For RNA-seq and microRNA-seq, we have the general, the regular things you would expect out of gene expression. Just gene expression files that were produced from those alignments. So because TCGA is such a large project and the data was inherited in different levels of processing, the GDC has some unique molecular data that came from TCGA. First, I want to bring attention to the copy number variation, for which we have these 2 files, the first being the segment files, which are generated by the R package DNA copy. And these come directly from the TCGA tangent copy number files. So these were tangent normalized using a specific algorithm. So it's not exactly raw data, but it is. But it was processed even more by the GDC using the GRCH38 coordinates. I just want to bring to everyone's attention that it's also available in a masked version that removes areas from the segments that are known to contain germline mutations. The copy number estimation files are a bit more processed. It's generated by the program GISTIC2, and it provides the copy number estimation at a gene level. And these files are available at the project level. So there are 33 of them, one for each TCGA project. Methylation data was calculated from the level 2 array intensities and then was remapped to the reference genome so that the coordinates would match everything else in the GDC. And then lots of metadata was added to each of these beta values regarding which genes they were near or in, that kind of thing. The attack-seq data that is hosted in the GDC right now is only alignment. So it's attack-seq data that was aligned to GRCH38. I just wanted to talk about some limitations. You see, the GDC is a living, breathing project, and lots of improvements are being made at the GDC at all times. So because TCGA was one of the first projects to be featured, it doesn't have some of the features that were developed later on. So this includes the alignment metrics, which you will find when you download alignments from more recent projects. And later on in the GDC's lifetime, there were there was an improvement in the RNA-seq pipeline that, in addition to the HT-seq counts, also included star counts, as well as splice junction quantification, and two additional alignments, one for the transcriptome and one for the chimeric mapped reads. The other alignment that's available everywhere is the genomic one, which is available across any project that has RNA-seq data. The good news is that this data will be made available in a future release when the TCGA data is reprocessed. This data will be reprocessed. It will match all of the other data. I don't have an exact estimate for when that will be, though. But this presentation is in February 2021, so in the future, this data may be available if you're watching this a while later. Okay. Now I just want to go in briefly for what happens to some of the variant data that is processed for TCGA. And that's this section of the flowcharts, the MAFs being processed into mutations. And in general, the GDC produces two different MAF files. One is raw and one is processed to remove lots of germline variants. If you watch the DNA-seq webinar, which is available on our website, you will they go into much deeper detail on how these files are produced. So anyways, these MAF files are indexed in the GDC, made available via the API from these open access MAF files. And this also, in indexing them, attaches a bunch of metadata to each of these mutations. So which patient case it's associated to, which gene it's associated with, and then clinical data, what consequences such a mutation might cause, and very basic information like the genomic coordinates. As you can see here in the screenshot, there's also things like survival data and some metrics about the mutations. I'm going to go into that in the demo section. Okay, and now I want to talk about the publication pages, which are produced when researchers process both TCGA data that they find on the GDC data portal and the original TCGA molecular data. And because the GDC inherited the TCGA program, the GDC hosts previous projects on previously generated manuscripts, ones that predate the GDC, on the publication page website. So the publication page data usually includes supplemental files that were generated by studies that use GDC, or in this case TCGA data, and usually these files are ones that don't fit into the GDC data model. So normally if a project produced a bunch of, let's say, molecular data, like a bunch of RNA-seq FASTA files, those would be those would go through the regular harmonization pipeline. If there was something like some tables that summarized results that were larger and maybe didn't fit in the manuscript itself, those would be a good candidate for files that would fit in a publication page. Like I was saying before, these publications also include data from older TCGA publications, one of which receives a lot of interest is the Cancer Atlas studies. So those studies have publication pages and associated files in the GDC publication pages.
Bill, I don't know if, I wanted to point out, I don't know if this was the time to do it, but just so folks know, there are a lot of maybe you're going to say it here. There are a lot of files that have some clinical data in it, and some genomic data that are associated with the publication pages. So just if folks aren't aware that there's a lot of great supplemental information, please be sure to check those files. Sometimes they are just spreadsheets on a case level, and I just want to throw that in there. But I didn't mean to interrupt, but I just wanted to, I didn't know you had another slide on it, but
Oh, thanks, Terry. No,
okay.
that's good information to give here. So, just some general logistics here. The files are downloadable either using links, which are you can just see in the blue here on the bottom. Those are the open access files, or all files can be downloaded using manifests that are provided on the publication pages. The manifests are downloaded into controlled versus open files and can be downloaded using the GDC data transfer tool. So before I leave this section, I just want to show these publication pages. So here's the GDC data portal, which most people, if they visit the GDC, here's what they visit. But the GDC publication pages are hosted on a separate site, which is the GDC website. However, there's a nice shortcut here in the portal, in which if you were to go to GDC apps in the corner here, click this button, and go to publication pages or publications, it brings you right to the page. So here's a list of the pages. Today we are interested in TCGA, so I will definitely click that. Everything can be filtered according to project, year, keywords, those kind of attributes. And in here we have the PanCan publications, and outside of this box here we have the non-PanCan Atlas TCGA publications. But clicking on each of these view publication information and supplementary files will bring you to the publication page itself with information about the citation, where to find the paper, and with links to both the manifests and some of the files, as well as instructions for downloading the data at the bottom. So continuing in this presentation, there's just a couple more TCGA resources that I'd like to bring to your attention, and I will just show them because they're worth simply showing than talking about and showing. So first, there's a few things in the GDC that are supplemental to TCGA that we want to make available. And in this case, one of them is the code table. So if you go down to the support page, click on resources for TCGA users, and click on the TCGA code tables, we have a list of different codes that can be used to interpret different pieces of TCGA clinical data, most notably the TCGA barcode. So if you wanted to know, for example, sample types, if you found an O1 in the sample section of the TCGA barcode, you would know that's a primary solid tumor. I will say that most of this data has been imported into the GDC as regular metadata, but this can be a nice shortcut if you're just going through lots of TCGA barcodes. So I won't even go back to the show. The next part I want to go through is a demo to show how to retrieve TCGA data from the GDC. So I'll start out at the portal, and the main place is the repository. That is the place to get files, and TCGA is no different. So clicking on this cases tab, you can go down to TCGA, and once that is clicked, all the TCGA projects, all 33 of them, are lined up here. As always, things can be filtered by clinical attributes or by file attributes, such as data type, experimental strategy, certain workflows, that kind of thing. And this specific function of the GDC is outlined in much better detail in other webinars, so please feel free to check that out or our documentation site for more details. Another place to find TCGA data is the exploration page. So if you were to filter things down by a certain set of files, or a set of cases. So for example, we want to look at TCGA lung adenocarcinoma. We would go to that, and view these cases and the exploration page. My apologies, I was doing some testing on a different environment, so I can show the exploration page. So here's the exploration page. It is functioning. That's just my fault for not clearing my cache. So we'll go back to the lung adenocarcinoma cases, and as you can see, we have the list of the most frequently mutated genes here, and it dynamically creates a survival plot for whichever set of cases you have selected. So yeah, there's just lots of features here. This is all described in other webinars and other documentation. I won't go too deep into this, but now I want to focus on how to find TCGA clinical data. So I'll go back to the repository, and I have a few things to show here. So if one thing you may notice is Tara talked about all of the wonderful attributes that clinical attributes that TCGA includes. And you'll see that there's not that many of them here on the side. And that's just because the GDC does not have room to put all of them. So if you don't see what you're looking to filter things down by here, you can use this function here, add a case biospecimen filter. If you click here, one of the most common examples is the sample type. Somebody might want to download only the alignments that are associated with primary tumors. So you can type sample type into this field here. This includes the sample type ID, but in our case, we just want sample type. So I will click on that, and I can search for only cases or only files that are associated directly to primary tumors. So I'll go back to only filtering for TCGA, and only filtering for TCGA lung adenocarcinoma. And all of the files here are connected to primary tumors. So if I wanted to only see RNA-seq files that are attached to primary tumors, here's how I would do it. So now I have this data set, RNA-seq files, lung adenocarcinoma, TCGA, and I want to know, I need clinical data for this. If you process molecular cancer data, you will more than likely need clinical data. So I'm going to show you a few methods for doing that. Number one is to add the files to the cart, go into the cart, and then choose this clinical button here. This will go through these 513 cases and give you just all of the clinical data that is indexed in the GDC. And when I say indexed, I mean those are the attributes, the properties that can be found through the GDC API, and kind of think of it as the attributes that show up in the GDC data dictionary. So it can be downloaded as both a JSON or a TSV. I'll show you the JSON version of this case. So this is everything for those 513 cases that is indexed in the GDC. So we have the case ID, diagnosis information, treatment information, and this is kind of layered as a JSON. If you wanted something more flat, you can download the TSV files, but those are actually separated out into types. So there's an exposure TSV, a demographic TSV, etc. Now let's say you go through that and perhaps you're looking for some data that that you're not finding that's indexed in the GDC. Maybe there's not a dictionary entry for it, maybe there is, but it's not populated. And the next place to check are the clinical supplement files. So this can be found by going and let's again filter down by TCGA LUAD and clicking on data category clinical. So if we look at the data format breakdown here, we'll see, and you could see on the right, this list of files here, most of these are BCR XMLs, like Tara was describing before. These are on a case level, a per patient level. So these might take a little bit of work to parse out. So each one XML per case. However, like Tara was also mentioning, there are what is called the BCR Bio tab, which is the third one down at the data format here. And when we click on it, we'll see that each of these for the lung adenocarcinoma project is described in this part of the file name. So if you wanted information about the pharmaceuticals, you would download the drug one. And if you wanted information about radiation therapy, you would download the radiation one. But the one I find to be most helpful is the patient bio tab. And this one has lots of clinical elements for patients. So I'll download it just to kind of show you what it looks like.
And just to jump in, Bill, that is the enrollment form that I talked about earlier. So that's the equivalent of the enrollment form.
Yes, this one is for the enrollment form. So this just looks like kind of a messy TSV if I open it in a text program, so I won't be doing that. But what I can do is open it in Excel here. And as you can see, it's parsed out pretty nice. It has information about gender, days to birth, and some things are not available, but it will tell you pretty clearly if they are or aren't. And there's some like more specific parts of the anatomical sites, like where it was found in the lung, and other information about staging and stuff like that. Some of this can also be found in the GDC API, but if you're looking for specific clinical attributes, it can't hurt to check the bio tab.
Yeah, I'll just jump in. I think it's good you pulled up this example. I don't know if this one is true, but if you, a lot of them, so I mentioned that we updated the follow-up form. We didn't go back and get new data from folks when we added things to the enrollment form, but it did happen on occasion. And you'll see in here how it says not available. It might be the case if we scroll down to newer cases that you'll see some data pop up. I don't know if it's the case for these specific attributes, but just a heads up that if you see a bunch of not available, make sure you look at the entire file, because sometimes you'll scroll down and see a subset of cases that have those data. And this might be a case where, so just another detail, and there's a million of them. I think this was a lung file. Is that right, Bill? Yeah.
Yes.
So there were some small differences between the way the lung adenocarcinoma and the lung squamous cell cases were collected. So you will see some small changes, even though they have one format for the lung files. So there's a lot of little nuances like that within the clinical data. So if you have a really specific question, please ask us. It will also help us to know what we need to update in our frequently asked questions. So don't feel like you have a dumb question if you have a question that you think is dumb. It probably isn't. It's probably something we should highlight somewhere within our documentation.
Cool. Thanks a lot, Tara. And that actually reminds me, I forgot to show we have another section on the gdc.cancer.gov website in which you can go to the FAQ section, and there's a special checkbox here for TCGA. So if you wanted to look through TCGA frequently asked questions, here they are. So yeah, something like, if you want to download TCGA archive files or have interest in TCGA methylation data, we have answers here. And we do update this based on questions we get to the help desk. So yeah, that's all I have for TCGA. As a reminder, the GDC does have a help desk, which can be reached at this address here. It can be found easily by Googling it, so feel free to do so. That reaches my team and I, and we're happy to answer any questions you have that maybe aren't covered here, maybe aren't covered in this webinar. Here's a link to the TCGA resources website. And if you have a lot of questions you might have about how to use the GDC can be answered on the GDC documentation website, which can be found here. Thank you all so much for attending this webinar, and I guess we'll go on to any questions we have.
Okay, great. Thanks, Bill. There's a couple questions. If folks can put their questions in the Q&A section, but we do have a couple of questions. The first is, what is the general depth of coverage for whole exome sequencing for FFPE samples?
Ooh, that's a good question. I don't know the answer to that, but feel free to follow up on the help desk, and we will contact our bioinformatics team, who should know this.
Okay. The second question is, do we have data from Asian cancer patients? And you might want to show how you can search in the portal for that.
Yes. So if you're talking about TCGA, you would filter down by TCGA. And then at the bottom here, there is a race field, and there are 685 Asian patients in TCGA. So, yeah, admittedly, it is a mostly European project, but there are people of Asian descent in TCGA.
Yeah, I will add a small caveat. This was self-identification, and there was no way to answer. I think in hindsight, this was a poor choice, but we went with the census, US Census Bureau answers, and there wasn't a mixed race option. So just keeping that in mind, that might not be 100% accurate. Although I think you can probably, guess that those that are marked Asian are Asian.
Okay, and then another question is there data from most commonly mutated genes for lung? And please demonstrate that.
Oh yeah, happy to demonstrate that. So if you're going to go to exploration, go to TCGA, and then select lung adenocarcinoma. And you could also add lung squamous cell to this set of cases. If you were to click on the gene tabs here, you can see that it was the most frequently mutated genes, right? They were asking for. I can't see the Q&A. Yes. Okay. Cool. Thank you. That just popped up for me. So here are the most frequently mutated genes. Of course, TP53 and TTN are the top ones. I know TTN is just a very large gene, so that sometimes brings in a lot of mutations. One thing that some people like to do is to only select by the cancer gene census genes. So if you were to select this, of course, TP53 would be top, but that removes things like the TTN gene and has more of a list that is relevant to known cancer genes, known oncogenes. Okay, are there any questions?
Yeah, can you expand on that and show how to get the complete list of genes, like actually how to download the list of genes?
Yeah, okay, so this is a good question. So there's 575 genes here, and you can download the JSON here. Now, if you were to unselect this, there is 21,000 genes, and this this will give you a pretty large JSON file. And this will give you the list of genes. So if you were to look at it, we do have the list of 21,000 genes here.
So Bill, do you want to show how you can save a gene set, that would then be used in analysis?
Sure. So if you wanted to save this gene set, this 21,000 genes, we actually couldn't do that right now because our limit's 10,000. But let's sort it down by the cancer gene census, and now it's 575. You could save this as a gene set, and it gives it a name here based on what filters you used, but you can name it whatever you want. So I can name this webinar filter, and I'll save it. And now I come back to this page later on without these filters here. And if I go to manage sets, I can look at this webinar filter here, change the names, and return directly to these same set of genes.
Okay. All right, thanks, Bill. There was a question on could you please link to any paper which applies machine learning techniques to this kind of data?
Ooh, that's a good question. I don't know exactly which. I don't know which papers those would be. I'm sure you could find them by going through Google Scholar. I don't know which of the GDC publication page will have that exact subject.
Yeah, there has been some work in using the images that we have on TCGA, the actual digital pathology images, using that for kind of artificial intelligence, looking at, can you predict survival or can you predict a tumor subtype? There has been that research going on that we know other folks are using TCGA data towards that. Okay. Will this presentation's recording be available in the portal? The presentation, the recording is available on our GDC website in the webinar. I did put that on the link to our webinar page in the chat earlier on so that you can kind of link to that. Just to note that it does take us some time to get it out there because we do have to make it compliant. We have to make sure, both the captions and the videos are properly edited. So it does take a little bit of time for that. Okay. Are there any other questions?
I see somebody has a question about the melanoma SKCM. Should I answer that?
Yeah, certainly, Tara. I don't see that.
Okay. I think I'm looking in the right place. It says, thank you for quite a specific question on TCGA SKCM. Sets the melanoma study. Days to sample has an interesting value for a number of patients, in some instances over 10,000 days, about 30 years. So melanoma, we did allow metastatic tumors to be submitted as the only tumor. And so you'll see some cases, this is a good point, the days 2 values within the data set are based on an index date of the initial pathologic diagnosis. And so for melanoma, that could have been many years prior to the sample that we received, because we did allow metastatic tumors to be submitted as the only tumor. For most of the other studies, in fact, I think all of the other studies with maybe a few exceptions, you had to submit the primary, and you could also submit a metastatic tumor, but you couldn't only submit a metastatic. And the reason we allowed that for melanoma is because for a lot of those cases, interferon was the only treatment that they received. And so, and a lot of them were handled surgically. And so we felt that the treatment would not interfere with their molecular profile. And so I think that's why you're seeing those. But if you have really specific examples that you want to send us, let us know. Oh, and you said that made sense. And then there was another question about the comment I made about self-identification on race. Yes, we took all of them to be self-identified. They came in from many, like I mentioned, many different organizations. But I think to be on the safe side, we considered all of them as self-identification, although we didn't, double check with every single site that that's how they classified it. Those are the two questions that I see that I can answer.
Tara, I just noticed my Q&A was frozen, the section on it. So there's other questions. I'm able to now access it. So
okay.
some TCGA files are not openly accessible in GDC. How does one get access to these?
Yeah, that's a good question. So data can be reached. This is done through dbGaP. So the general flow for this is um, to first get an ERA Commons ID, which is the one you will use to log into the GDC. You can then use the ERA Commons ID to log into dbGaP, in which you can apply for the TCGA project. Applying for the TCGA project, you will need to know the code to apply for it, and that code we can find here is PHS000178. This is the ovarian project, but this applies to all TCGA projects. So once you get access to PHS000178, you will have access to all controlled data across all TCGA projects.
Thanks, Bill. And this goes along with the other questions are what are controlled data sets and how do we access patient-specific DNA sequence data of a set of genes? And so it's kind of all related to access.
Yeah, so if you go down and you click and you filter by either controlled access or by open access, you will see that the controlled data, and I'll filter down by TCGA just so that we're looking at that project. But in general, the rules are pretty similar across GDC. If it contains DNA sequence data and it can potentially allow for a patient to be identified, that is always controlled data. So all alignments, all VCFs are controlled, and so VCFs and BAMs are always controlled. A subset of the maps are controlled, but there are a subset that are open, and they are the one exception to the sequence data, since you will see, which positions have which DNA changes, but the open access maps have a large pipeline that they go through to verify, a very conservative pipeline to make sure that no germline mutations make it into the open access map.
Okay, here's another question about TCGA SKCM. Days to sample has interesting values for number of patients. In some instances, over 10,000 days, which is approximately 30 years.
That's the one I just answered.
Yeah. Okay. I'm sorry.
You're fine. And I answered the next one too.
Yeah. Okay. There's if it's possible to automate TCGA data extraction via an API.
Yes. TCGA data can be retrieved via the API. I don't think I have time to get into exactly how to do that, but we do have a webinar that goes through how to use the API. This was done a while ago. GDC API and apps. And also, if you go to the documentation site, go to API at the top here, and click on Search and Retrieval. This will give you information on how to put together queries to retrieve TCGA data. If you want to know how to download the actual files through the API, we have another section on downloading files. And as always, if you have any questions about the API, feel free to contact the help desk. We are happy to help with that.
Okay, are there VCF with germline variants available for the DNA data? What's the best way to find it?
So the GDC doesn't do any purposeful germline calls. We know that, when you do somatic variant calls, which is what the GDC does primarily, some germline mutations may make it through. So if you have access to the VCFs, you might be able to check those out. In general, if you want a comprehensive list of germline mutations, we would suggest you download the BAM files and do the calls yourself.
How do you access TCGA FASTQ files?
TCGA FASTQ files are not available exactly in that way in the GDC. However, within these BAM files, this includes the aligned and unaligned reads. So if you extract the reads from this, you will have what is effectively the original FASTQ file. So I think you can use, most people use SAM tools for that, I think, but there are some other programs that can do the same thing.
Going between the chat and the QA, and I think there's one question in the chat. Is the data available to non-US citizens? Or they're a professor from Brazil?
Yes. So if you go to portal, so right now I'll just go to portal.gdc.cancer.gov. This doesn't require any sort of login. It doesn't require anything. I can go to the repository directly without logging in or anything. In terms of controlled access data, I know that ERA Commons and dbGaP doesn't it's not limited to US citizens. I know groups from other countries that are not the US get access all the time. I don't know, I can't tell you if there's any specific limitations on Brazil or anything, but I know that it is not just limited to US citizens.
Okay, I think that was I think we covered all the questions. Does anybody have any other questions? I'm looking at those. Okay, I think we're also out of time as well. Thanks, Bill and Tara, for this wonderful presentation and the demonstration. And thanks to the participants for attending today's meeting. Please, watch the support webinar page for the video. We will be posting that in the future, and we do tweet about our available videos as well. But thank you so much, everybody, for attending. Have a good day, everyone.
Thank you, everyone.
Yeah, thanks.